Calling you. This is the student day, welcome to MouShauk. And we will start today with a new chapter on biology of class science syllabus, that is economic importance of bacteria and fungi. Yes. So before I start, I will request you to please to subscribe my channel if you are not subscribed yet, or if you are a new viewer to the channel. Okay? And if you are liking the video, press the like button, share the video with friends so that they can also be benefited from this video. And do meditate before we start. So before we start our discussion, we'll have a short presentation session, and then we will start with the discussion. Okay? So for meditation, please concentrate on one slide. The slide is starting with you. We are when we study this economic importance of bacteria and fungi, we'll have a short gist on this chapter. That is, what topics we will discuss in this chapter, and then we will come to its importance. What is the importance of this chapter? Okay. So, for bacteria, economic importance for bacteria, we will study a general study of the bacteria. Then what is its role, useful role in medicine? What is its role in agriculture? What is its role in industry? And some harmful effects of bacteria, that is like spoilage of food, the diseases in plants and animals which it spreads, and then bioweapons. Okay? And in case of fungi, we will discuss here also we will discuss a general study on the fungi, then its economic importance of yeast. Yeast is a fungi. Its useful and harmful effects, or rather useful and harmful fungi we will discuss. Useful role of fungi and what is its role in yeast. Okay, now this economic importance, why do we need to study this? Excuse me. Why do we need to study the economic importance of this bacteria and fungi? The bacteria and fungi are okay. So what is their importance? So bacteria are extensively used to manufacture different nutritional supplements and many medications. So, for example, see E. coli. E. coli is a bacteria which is used in the production of vitamin K and riboflavin, and also it is used to make products like vitamin D, amino acids. Okay. Then providing a key precursor in amoxicillin production, which is an antibiotic. Even in fungi is also important for organisms in human life. It's an important organism for human life. Okay. They play an important role in medicines, with antibiotics are also used for fungi. Then in agriculture, by maintaining soil fertility, then they are consumed as food. They form the basis of many industries. Okay, so all these are important of this bacteria and fungi. And to know this, to know all these in details, we need to study this chapter. Okay, so for this, first we need to study their general characteristics of this bacteria and fungi. And then we will come to the importance. Okay. So first we come to the general study of the bacteria, the characteristics, features of this bacteria. They are generally most primitive unicellular organisms, and each cell contains a single chromosome, and that is not enclosed in a nuclear membrane. Okay, they are prokaryotes. They are, that's why they are not plants. Okay, they are not considered as plants because they are prokaryotic. Okay, that is their nuclear material is very pure, okay? And they are not under any bounded nuclear membrane. Several thousands of known species of this bacteria are known. Then they occur in air, water, soil, food, or on inside the body of living organisms, practically everywhere they are. Okay. And in human body, they are found in mouth and skin, inside the intestine, in the genital tract, everywhere. Okay. And in generally, uh, this when they are lying on the skin or intestine, in genital tract, some are useful, but some are harmful also for the diseases. So we'll discuss later on. Now, some intestinal bacteria are beneficial, synthesize vitamin D. Some harmful bacteria that cause diseases also. Some useful ones are prevent the growth of the harmful ones. There are some useful bacteria which prevent the growth of the harmful ones. Since they are the smallest living organisms on the earth, average size is 2 micrometers long and 0.5 micrometers thick. Okay, their average size is 2 micrometers long, length is 2 micrometers, and their thickness is 0.5 micrometers. Now their shapes. Generally, there are 4 shapes. One is puffy, capsuli, spirilla, and bite. So puffy, they are spherical. Capsuli, they are broad-shaped. Spirilla, they are spiral or twisted. Vibrio is comma shape. It's the comma. We put a comma in English alphabet, in English sentences. So punctuation marks, those shapes that comma shape that we now, many of them they live singly, but some occur in pairs, like Diplococcus. Then they can appear in long chains, like Streptococcus, or they occur in clusters, Staphylococcus. So, Diplococcus in pairs, long chains, Streptococcus, clusters, Staphylococcus. Okay? Now, their structure. As I told you, their nucleus is very primitive, prokaryotic, that is, their nucleus is not bounded by a nuclear membrane. So the cell should also be a very simple cell, very primitive one. That is, their cell is a primitive cell, a simple cell, containing protoplasm covered by a hard cell wall, which is made up of peptidoglycan and not any cellular wall. The cell wall is made up of peptidoglycan. Immediately below the cell wall lies the cell or the plasma membrane. Okay. Cell wall, we know that it is permeable, but cell membrane or plasma membrane, that is semi-permeable, allows very selective substances to get inside the cell or outside the cell. Then they do not have any well-defined nucleus. DNA or chromatin material is specially placed in the form of a single circle. Okay. Sometimes a slimy protective layer capsule surrounds the cell wall. Under unfavorable conditions, the capsule covers the slimy protective layer which forms around the cell wall, protecting the bacteria from this harsh conditions and it forms the spore bubble. Okay. Now, no membrane bound stem organelles are present. Excuse me. Vacuoles, granules, and sometimes chlorophylls are present. So the structure of the bacterial cell is like this. See, there's the capsule layer, dark brown color layer, that is the capsule layer, below which lies the cell wall, and below that is the plasma of the cell membrane. The cytoplasm is there, okay, which contains many glycogen granules, vacuoles, chlorophyll substance, chlorophylls are also present. Okay. Then DNA material is placed in the center of the cell, forming a circular structure. Okay. Then some plasmid are present. Okay. I'll talk about what is plasmid later on. It's coming up. Okay. Then what else? Yeah, flagellum is present. Okay. Flagellum, these are substances, these are hair-like structures which help them to move about. Now we come to the movement. Some bacteria are non-flagellative, but those non-flagellative bacteria, they cannot move alone. They have to be transported by wind, water, or any other external source. Okay? But flagellated bacteria, they move by lashing movement of the whip-like flagella, one or more whip-like flagella, piercing through the cell wall or the capsule. These flagella, they come out of the piercing the cell wall of the capsule and the capsule. They're whip-like structures, and with the help of these whip-like structures, they move. They can move. So the lashing movement of the flagella provides active locomotion in a liquid environment. When this bacteria, this flagellated bacteria, they are in liquid environment, they help this bacteria to locomote from one place to another, to move from one place to another by moving their flagella. Now, the nutrition. Most of them do not have chlorophyll, so they are not autotrophic mainly. Mainly, they are heterotrophic, that is saprophytic and parasitic, or parasitic. Saprophytic or parasitic. Now, saprotrophic or parasitic, saprotrophic means that they depend on dead organisms, and parasitic is they depend on living organisms. Then they secrete powerful enzymes from their cells into the surrounding food material, which makes the food material soluble and is then absorbed as a solution into the cell. Okay, so what happens? The food material, which is very near to the bacteria, they will secrete some enzymes, powerful enzymes from their body, which will surround the food, which will liquidize the food into a soluble form, and then the bacteria will absorb this soluble food into the cell. Okay. Now, respiration. Respiration is they respire both aerobically and anaerobically. Okay. Now, anaerobic bacteria, they will be killed if they are exposed to. So they aerobically or anaerobically they will respond. Now we come to reproduction. Mainly they have a asexual reproduction. Okay, that is through by binary fission they divide. Cell division occurs through binary fission. That is only under favorable conditions. Okay. During this binary fission, DNA elongates. Okay, and it will be DNA elongates and will be followed by a transverse constriction from the appearing in the middle of the cell, and the cell divides into two cells. Depending on the type of the bacteria, the two daughter cells will remain attached or get separated. Depending on the bacteria, whether they are in Staphylococcus or Diplococcus or Streptococcus, according to their bacteria type, they will get independent or they will remain attached, the daughter cells. Now, under unfavorable conditions, what happens? The protoplasm forms a spherical mass. It forms a spherical mass which receives a thick impervious wall around it. Okay, it forms a thick impervious wall around it, which is called a spore. Okay, and this spore can remain alive. The bacteria inside this spore, inside this impervious wall, they will remain alive. Okay. And when the favorable condition returns, the wall breaks, making the bacteria, which helps the bacteria to come out. Okay. The spores can tolerate extreme dryness, extreme temperature, and can tolerate poisonous chemicals. That's why the bacteria can remain alive inside that spore wall, impervious wall. Now, there's a picture of the reproduction in the bacteria by binary fission. See, the cell has this nuclear material inside it. Now, this nuclear material elongates and it duplicates, DNA duplicates, and it makes a copy of it. And then there is a constriction occurring in the middle of the two sides of it, and it separates into two daughter cells, having the chaining material in it. So that is the reproduction of bacteria by plant. Now, this is the spore formation. Okay, under unfavorable conditions, I told you that this bacteria that forms the wall around it, bacterial wall, and the spore wall is there also. It will remain inside this spore wall. Okay, and it will remain in when favorable conditions occurs. This spore wall breaks, the bacterial wall and spore wall breaks, and the spores comes out and germinates. Okay, and it starts becomes active. Now, besides this asexual reproduction in bacteria, very few of them, they have highly primitive sexual type of reproduction. Okay. In this, two bacteria of the same species but of different strains, they conjugate. Okay. When a small circular DNA molecule that is different from the chromosomal DNA, that is known as plasmid. So what is plasmid? Plasmid is a small circular DNA which is different from the usual chromosomal DNA. That is known as plasmid. That plasmid, what happens? That gets transferred from the donor to the recipient through a hollow tube-like extension of the donor cell. The donor cell forms a hollow tube-like extension to the recipient. And through this tube-like extension, the plasmid gets transferred from the donor to the recipient. This is how sexual reproduction occurs in some of the bacteria. Not all. Generally, the sexual reproduction does not occur in bacteria, but in some cases, this sexual reproduction occurs. Okay. Mainly, generally, the asexual reproduction. So this is how the general study appears to us. Now we will come to the economic importance of the bacteria in different fields, especially in medicine, agriculture, industry, and some harmful effects of bacteria. Okay, so we will discuss in my next video. So till then, please go to this. If you have any doubts, please let me know. Write in the comment box. You can also write in my email ID, that is kasturi74@gmail.com. KASTURI kasturi74@gmail.com. Please write here. And If you are liking the video, press the like button, share with your friends so that they can also be benefited from these videos. And do meditate before you start your day and do maintain a chart. Okay, so that you can complete according to your chart the whole day. You can monitorize how much you have completed, how much is left, and how do you proceed for the next day. Okay, so this will help you to prepare in your exam. Okay, so till then, please go through this and have a nice day. Thank you. Thank you for joining me.